Okay, it's time for the next speaker, that is Andrew Bowman. He is a senior scientist at AbbVie. His research is focused on mass spec imaging for pharmaceutical applications, and especially in drug metabolism and deposition. So, welcome, Andrew. Both of them.
Let's see, does that work?
Yeah. Thank you.
So thank you everyone for coming. It's still this early in the morning. I don't know how many of you are morning people, but I'm not, so I apologize in advance. I will actually be able to skip through a fair amount of this because Professor Patty did me the honor of covering a lot of the things that I was going to cover real quickly in this talk. So I am at a large pharmaceutical, so everyone gets to see this nice disclosure information to say that I am paid by AbbVie, so anything I say about AbbVie products is suspect. But what I'm really going to show you guys here is the specific applications that I, as a man in pharma, am using mass spec imaging for, which is generally a targeted approach to mass spec imaging. And so for pharma as a whole industry, one of the things that we are very used to doing is looking at drugs for pharmacokinetics, pharmacodynamics, trying to look into pharmaceutics and pharmacology from the perspective of looking at matrices. We're looking at blood, we're looking at urine, we're looking at feces, we're looking at bile, and we're trying to figure out what the drug is, what it's becoming, how much of it is turning into other things, where it's going. And then for some of those other sections where we're like, it's not really staying in the blood, where is it going? Is it getting into the tissue of interest? And so we'll take a liver, a kidney, a brain, a tumor, and we'll homogenize it so that we can get something that we can put into an LCMS. Now that tissue homogenization process, really great, gives you lovely sensitivity, but the problem is that it sort of fails to distinguish that a tissue is a very complex beast. It basically says everything in this tissue is exactly the same, whether I'm looking at a bile duct or I'm looking at a hepatocyte. That's just all liver. It's fine. And so you can often miss very subtle changes or very gross molecular changes just because they happen only in a very small percentage of cells. So one of the challenges that I was brought into AbbVie to address whenever I was a postdoc with them was being able to do tissue imaging for these low abundance compounds to be able to look for them and say, is our drug getting to the place that we think it is? And additionally, we started looking into triple quadruples because we needed to look at the quantitative biodistribution of drugs. Because for a lot of our project teams, if the data isn't quantitative, it's useless. So being able to quantitate in a mass spec imaging format becomes very important. So Professor Patty covered basically everything you guys need to know about mass spec imaging, but as a general rule for broad spectrum analysis, you're able to look at everything that's in a tissue, or at least everything that you can ionize in a tissue. So that's everything that's desorbed from the discrete array of spots, and then the abundance of those individual ions are plotted, and you kind of hope that they are either close to what is in the endogenous tissue, or you guess that they are. But as Professor Patty showed off, that assumption might not always hold. But one of the last things that we like to be able to do with our tissues is compare compound distribution to histology results. So we often correlate our data with either H&E, fluorescence, or MRI, or a bunch of other imaging technologies to say whether or not the things we're seeing match with, say, the same things that people are seeing if they take these drugs and they do positive emission tomography on them. So we like to call it in AbbVie molecular histology, MSI combining that high sensitivity and accuracy of mass spectrometry for the identification with histology-like information, the spatial distribution within tissue sections. And so we have started to use this in a broad variety of aspects at AbbVie. One of the most important ones for us is target engagement. Is the drug reaching its target? Are we able to determine any level of target occupancy? Can we tell how long it's the target? And this helps with like compound selection, formulation. We've also done a fair amount of work at AbbVie for biomarker identification, looking for markers of efficacy, explores the signaling mechanisms, which helps to understand the pathology to help discover therapeutic insights. For multimodal integration, we are, of course, the MassTech imaging group at AbbVie, but we like to be able to relate this to the H&E stains, IHC, ISH. We've worked with a variety of other techniques on the same and adjacent tissue slides. But this helps with like hepatotoxicity, CNS penetration, looks for tumor distribution and PK-PD relationships. We've also done a fair amount of work with toxicity support, which is helpful for safety. Of course, just looking at the tissue distribution of the drug in general, as well as any biomarkers that we see around it. And we're starting to use this technology now in actual human clinical trials. So whenever you're looking at MS imaging versus other sorts of technologies or other forms of mass spectrometry, the spatially resolved molecular information across the tissue, it's a label-free analysis. It can detect a wide range of molecules with pretty good sensitivity and specificity. You can detect thousands of compounds all at once, and that combined with MSMS information can provide structural information. Unfortunately, whenever you're dealing with a lot of samples, and I think everyone who has ever had to deal with sectioning either an entire zebrafish or hundreds upon hundreds of brain tissue slices can tell you that they generate large, complex data sets that require very advanced analysis techniques to be able to actually understand what you're looking at. Often they require meticulous preparation, and so if you do that preparation wrong, it greatly affects the quality of the data that you're able to generate. Quantification can be a great challenge because different tissues have different effects on the drug of interest. And then often that instrumentation is costly and very complex. So because we are mostly interested for the kinds of studies I'm doing in sensitivity and specificity, I'm looking for my drug of interest. And because I have to do this a lot and I don't necessarily want my instrument to be big and complex, moving from a broad spectrum analysis to a targeted analysis allows me to move to instruments that are less complex, less costly, but are more sensitive. So moving from untargeted to targeted imaging means that there are advantages to untargeted imaging. You can do spatial mapping. And this is very ideal for exploratory kinds of studies where I don't know exactly what I'm looking for. I don't know if my drug is making different kinds of metabolites. So I'm just interested in finding out as much as I can. But those are those big, large, complex data sets. They're generally only semi-quantitative at best, and they generally have less sensitivity for many of the compounds of interest. So this is what we're often using for studies that are focused on like identifying or discovering molecules. So if we're looking at a new disease state that we've never seen before, we don't know exactly what we're looking for. So I just kind of need to run healthy and diseased and say, what's different? And then this also is helpful for looking at broad metabolic pathways across tissues. By comparison, looking at targeted mass spec imaging, this is for high sensitivity, high specificity. This allows for more accurate quantification, and it's a very simplified data analysis. Instead of looking at a data set and going, here's 1,000 things that I don't know what I'm looking at, I can now go, here's 6 things that I'm reasonably sure about. But because I'm only looking at 6 things, I can only analyze those 6 things. Everything is predetermined, and so there's a lack of exploratory potential in being able to do targeted imaging analysis. So this is the kind of technology that's best suited to detailed investigations of known compounds, looking for target engagement, or monitoring specific drug metabolite distributions across tissues. So the crux of this is that there is currently not a targeted MALDI platform that is commercially available that combines sort of this ability for high spatial resolution with a highly robust workflow and a user-friendly experience. So AbbVie, in partnership with both Agilent and MassTech, worked on really getting this targeted imaging process up and running at AbbVie. And so this was focused on improving our sensitivity for our drug efficacy, biomarker monitoring, or tissue PK studies. So we're looking for compounds that are often very low dosed, or their ADC moiety payloads where they are dosed at a milligram per kilogram for an animal, and then of that, the actual compound of interest that I'm looking at is 0.1% of all of that. And this is what we're using for quantitation. So Agilent provided us with a 6495D, which is a state-of-the-art triple quad. They're also providing us with strategies to obtain the best sensitivity and data quality using their instrument. And they're also providing some support for data processing and some support for the hardware modifications. MassTech provided the AP-MALDI source, as well as the ability to modify the inlet of the system. And then AbbVie, read me in this case, since I've done most of the work for this at AbbVie. I'm getting samples from other groups at AbbVie, but I'm the one who's doing all of the work on running it. I'm providing feedback to both Agilent and MassTech about scientific gaps in the robustness of this system and provide ideas for enhancing the technology. So Professor Patty showed off everything you need to know about triple quads and why we were moving to this fragment selection. But that removal of noise is really what we're looking for to improve our sensitivity. And what that kind of looks like in practice is that whenever we move from a timsTOF flex, doing sort of a quick image analysis, if I'm looking at MMAE, which is a drug that is often attached to antibodies, the drug plus the linker, I can get up to 170-fold better limits of detection by using a targeted imaging platform. If I'm looking at just MMAE itself, I can still get 16-fold better limits of detection. And because I always like to show off that there's a failure state for everything, reserpine, which is that fun compound that everyone likes to use to talk about how sensitive their equipment is, it looked exactly the same to me between the timsTOF Flex and the Agilent 6495D whenever you're imaging it. But despite having a couple of failures like reserpine, what it looks like whenever you start to need that level of sensitivity, this is dosed mouse brain. On the left is what I ran for the timsTOF Flex. This was an untargeted approach. That has been dosed. You will note that there is not a lot going on in there that I can tell from the timsTOF Flex. It looks awfully like what it looks like on the triple quad whenever I am looking at the vehicle, and that there is nothing, which is great. I want nothing in the middle, but I should be seeing something on the left. But if I run this exact same experiment on a dosed tissue on the right, all of a sudden I can start to see different brain structures where this drug that I know penetrates the blood-brain barrier is actually very well distributed throughout the brain. The last couple of slides in here are me picking a fight with Eugene specifically about this, because he told me whenever we installed this that I didn't need to use 10 kilohertz for this laser. I could move all the way down to 1 kilohertz and still get good data. But what I found is that if I adjusted the laser frequency, the faster I ran the laser, the more data I got. And so if you go from 1 kilohertz to 5 kilohertz to 10 kilohertz, you start to see better and better representation of the data. But there is also sort of this funny thing where I had been running some of these experiments on a SciAx 6500 Plus with an MassTech AP-MALDI source, and I've been running all those 20 to 30 milliseconds for the transition time. And what I actually started to find is that as you decreased the accumulation time for these, I actually started to get better sensitivity. And so I have some ideas for why that is, but for those of you who are interested in doing triple quadruple work, it is important to note that there is, in fact, a sweet spot for the accumulation time for your analytes of interest, and it's maybe not as long as you think that it should be. And then sort of the same exact results. This is looking instead of at the endogenous compound and homogenate, this is spotted reserpine on top. And that just sort of recapitulates that I see the exact same thing with something that is in a smaller location that I've dosed on top of this. The faster I run the laser, the more sensitivity I get. And the shorter I run the accumulation time, the better sensitivity I get. I will still note that by the time you get down to 1 millisecond, you start to get into this thing where the data becomes less stable. So it looks more sensitive, but also the relative standard deviation is significantly higher at 1 millisecond than it is at 5. So 5 milliseconds is usually about where I keep it for the 4 to 6 transitions that I'm usually running in an experiment. But with that, I was just kind of showing you guys some targeted imaging work, specifying that there is a growing need for high throughput targeted mass spec imaging, that there is a great potential in combining AP-MALDI with triple quad to serve that purpose, and that we are still developing this technology. And with that, I'm happy to take any questions.